Okay, let's continue. I'm sorry for the delay because there is a technical issue. Okay, so this is the process that shows the gram staining process. Okay, so I'm sure that you have done this in the lab practical. Okay, so first of all, we have to heat fix, or we have to dry the slides, and then we put the crystal violet stain. Okay, so the color of the crystal violet, so is in the violet, or is something purple. And then after that, we put the iodine fix. Okay, so at this stage, the color of the cells are still using the color of the crystal violet stain, so which is purplish color. Okay, and then after that, we will destain the cell or the bacteria with the alcohol or with the acetone. Okay, at this stage, the gram-negative bacteria will decolorize. Okay, so the color will be colorless, but then for the gram-positive, it will remain the color of that crystal violet and also the iodine. The next one, we will stain the slide with the saffron color, and because the gram-negative bacteria is already decolorized from the previous step, then it will take up the color of the saffronian, which is the pink color. But for gram positive, because it remains the color of the previous stains, which are the crystal violet and also the iodine, so the saffronian color will not appear in gram positive slide. So this is how we can differentiate the bacteria, whether they are gram negative or the gram positive, based on the color, because the gram-negative bacteria, it will appear pinkish, and for gram-positive, it will appear purplish. Okay, so this is the same process that I have mentioned you just now in the previous slide, but it also has the explanation on what actually happened on each step. Okay, so the first step is on the heat fix. So why we heat fix, or why we dry the slides, so that the bacterial cells are fixed to the slide, so they can't run away or they can't move. And then after that, we flood the slide with the crystal violet dye. Okay, so at this point, the crystal violet will penetrate the peptidone glycan layer of both gram positive and gram negative. So that's why both cells or both bacteria types, they are colored with the crystal violet dye. Next one, we flood the slide with the iodine. Okay, at this stage, it has a different reactions to the gram positive and also the gram negative bacteria. So for the gram positive, the iodine ions, it will penetrate the cell wall, and it will bind with the crystal violet from the previous step. However, for the gram-negative bacteria, it has a reaction between the iodine ions and also the crystal violet, so they form a complex. So we call it a crystal violet iodine complex, and it's very big, and also it is insoluble in water. Next one, when we decolorize the slides with the alcohol or the acetone, it also has different reactions to the gram bacteria, gram positive and also the gram negative. Okay, so for the gram positive, um, if you still remember, the gram-positive bacteria, it has a thick peptidoglycan layer. It serves as the protection. So although we flood the slide with the alcohol, the peptidoglycan layer is protecting the cell from burst, and is protecting the cell from the it's protecting the cell, so that the crystal violet iodine complex cannot go out of the cell. So that's why at this stage, the gram-positive bacteria, they still remain the color of that crystal violet iodine, which is purplish. However, for the gram-negative bacteria, when we put the alcohol, when we stain the cell with the alcohol, it will disrupt the cell membrane. So that's why the crystal violet and also iodine complex, they can get out of that cell. Why it is disrupted, not the gram positive disrupted? Because going back to the bacterial composition of the gram negative, they have a thin peptidoglycan layer. Okay, so remember, peptidoglycan layer, it serves as a protection to the cell. So since gram-negative layer, gram-negative bacteria, it has a very thin peptidoglycan layer. So that's why it can't really protect the cell from the alcohol. So it bursts out, and the big crystal violet iodine complex is able to exit the cell. So that's why at this stage, the gram-positive bacteria, they remain color, the purplish color from the crystal violet and crystal violet iodine complex. However, for the gram-negative bacteria, because of that crystal violet iodine complex leave the cell, then it becomes colorless. The next one, we flood the cell with safranin dye. Okay, so safranin for the gram-positive bacteria, so safranin is still penetrates into the cell. However, because the gram-positive bacteria too, it still has that crystal violet iodine complex, then that complex is overpowering the safranin dye. Okay, so that's why we can't visualize the color of that safranin on gram-positive bacteria. So the color of gram-positive bacteria remains the color of that crystal violet iodine complex. On the other hand, for gram-negative bacteria, because it is colorless, right? Because it has been decolorized by the alcohol from the previous step, then when we flood the cell with the safranin, then it will be the color of pinkish. Okay, and then after this step, safranin is the last step. Then there is the next step is for the microscope. So that's why under the microscope we will be able to see that gram positive, it has the purple color, and gram negative, it has the pink color. Besides gram staining, to know the group of gram positive and also gram negative, there are also other stainings for other types of bacteria, or if you want to look at other composition of the bacteria. Okay, so for example, acid fast staining. So this acid fast staining, we use it to stain bacteria with high lipid and also high wax content. Next one is acridine orange. So this is when we want to locate the bacteria in blood cultures. Then there is the methylene blue stain. This is to stain for a very specific bacteria. It's called the Coryn bacterium diphtheriae. Lactophenol cotton blue and also the cocoa flower white. It is for fungal stain. And we also have the India ink stain. Okay, so this is the stain for bacteria with capsules and or for yeast. Okay, all right. So when we want to identify them through colony morphology and also the gram staining, after the gram staining process, we can look the slides under the microscope, and first of all, we can see the color. Okay, so from the color, we know that whether it is gram positive or the gram negative. The second one is we want to look at the shape of that cells, whether it is cocci or bacilli or spirochetes. Okay? So cocci is the spherical shape, or bacilli is a rod shape, and spirochetes is the helical or is worm-like. Okay, so that is one thing that we can take note as well to identify the bacteria. So besides the shape, we can also take note on the groupings of that cells, whether they are single or in pair or in clusters or in chains or in palisading arrangement. Okay, so those are the informations that can be used or very useful for us to identify the type of bacteria that we have. So besides that, we can also look at the size and also the length, whether it is short, long, filamentous, or curved, and so forth. Okay, there is another structure on the cell that we can also look to identify the bacteria. So it is called the cell appendages. And for the cell appendages, there are two types of structure that we can look on. So the first one is on the flagella. So flagella is a structure that helps the bacteria to move. Okay, so the second structure is the pili or the fimbriae. So fimbriae is the hair-like objections where it helps the bacteria to attach to the surface. Okay, so remember flagella, it helps the bacteria to move, and pili, it helps the bacteria to attach to the surface. So examples of the flagella. Okay, so flagella, remember, it helps the bacteria to move. Okay, it can be three different structures. Okay, so the first one, the flagella can be on only one end of the bacteria, and that is called the monopolar flagellation. Second type of flagella, it can be on both ends of the bacteria, and that is called the bipolar flagellation. And the third one, when the flagella is everywhere surrounding the cell, and that is what we call the peritrichous flagellation. Okay, so this is the figure. It shows about the flagella and also the pili. Okay, so remember, flagella is for movement, pili is for to attach to the surface. Okay, and the length of these two structures are different as well, because flagella, it helps in the movement of the bacteria. That it means that it has to be thicker and stronger and longer. But for pili, because it is for the attachment to the surface, then it has to be a lot on the surface of that bacteria. It doesn't that long for okay, so let's look at bacteria genera. Okay, so for example, Escherichia. So it is very common to be found in the human intestines, and it can cause very serious foodborne disease. Okay, so for example, the E. coli. Okay, second one is on the Salmonella type. So Salmonella type ni, usually it can be found in the meat and poultry or contamination. When the meat and poultry are contaminated, and because the Salmonella type is a very potential pathogens. And when we talk about pathogens, so it means that it is a bacteria that cause severe diseases. Okay, so next one is the Serratia. Okay, so Serratia usually can be found on the catheters and in saline irrigation solutions. Okay, and the last one is the Proteus. And Proteus is very motile. It moves very quickly. Is everywhere because it has a lot of flagella, and it can cause the UTI, the urinary tract infection, or the wound infections. Okay, so that's all for chapter 3, part 1. We will continue with the next part. Thank you.